Great. Thank you, Marie, and thanks to all of you for joining this webinar series. I want to recognize very readily our collaborative support from ASN for this webinar. I'll note as we go to the next slide that ILSI North America is a nonprofit scientific organization that advances food safety and nutrition science for the benefit of public health. We do not lobby or develop policy positions. Rather, what we do is we bring together scientists, such as those of you on this webinar, from government, industry, and academia to tackle areas of great interest. While a lot of what we do is focused on science, I'm going to also note that we are very proud of our recognition of being platinum rated by GuideStar. We received the Platinum Seal of Transparency from GuideStar. Only 1% or so of all nonprofits have that transparency ranking. Why am I bringing this up? I'm bringing this up because within ILSI North America, our core values include public benefit, collaboration, transparency, and scientific integrity. We earned that platinum ranking because we could demonstrate not only that our mission and vision was towards public good, but because we could demonstrate our metrics on how we were advancing causes. So with that, want to again say welcome to the webinar, and Marie, I'll pass it back to you.
And with that, I am quite pleased to introduce Dr. Liping Zhao. He is currently the Everly Fenton Chair of Applied Microbiology in the School of Environmental and Biological Sciences at Rutgers University. He is the director of the Center for Nutrition, Microbiome, and Health of the New Jersey Institute for Food, Nutrition, and Health at Rutgers. Dr. Zhao is a fellow of the American Academy of Microbiology, and he's a senior fellow of the Canadian Institute for Advanced Research. He serves on the scientific advisory board for the Center for Microbiome Research and Education of the American Gastroenterological Association. Dr. Zhao's team has pioneered the approach of applying metagenomics, metabolomics, integrated tools, and dietary intervention for systems understanding and predictive manipulation of gut microbiota to improve human metabolic health. The clinical trials of his group have shown that dietary fiber modulation of gut microbiota can significantly alleviate metabolic diseases, and examples include a genetic form of obesity in children, as well as type 2 diabetes in adults. Science Magazine, interestingly, featured a story on how Dr. Zhao combines traditional Chinese medicine and the study of gut microbiota to understand and fight obesity. So with that, I would like to turn the podium over to Dr. Zhao to share his information with us, and we greatly thank him for taking the time to share his insights today. So Dr. Zhao.
Thank you, Marie. Thank you, Dr. Jones. Hi, everyone. I'm really grateful that ILSI North America and American Society for Nutrition can host this webinar for me, and so I can share my research on how nutrition, particularly dietary fibers, modulate gut microbiota and improve human health. And we know that the total complement of microbes living in and on human body is called the human microbiome. And the diversity and the density of microbes living in our gut is such that we can call it our microbial Amazon jungle. And the reason these bacteria are important in human health is that when they grow and maintain a certain population level in our gut, they produce potentially bioactive compounds, being beneficial or detrimental And those compounds can get into our bloodstream, circulate through our body, and interact with our own genomes so that they make a contribution to the development of our phenotypes. There have been so many studies now on comparing the gut microbiota between diseased people and healthy people, and they show difference. However, the causality question still remains for most of the cases. Is it because I'm diseased so that I have a different gut microbiota? Or it is because I have a different gut microbiota so that I'm diseased? Chicken or egg, which comes first? This is the fundamental question in all the microbiome-related research. And in this Nature Reviews Microbiology Perspective article, I argue that we still need to follow Co-occurrence postulates and to answer the causality question for microbiome research. But we need to modify and adapt to the polymicrobial nature of microbiome study, and also the two-way street nature of the microbiome study. So that means instead of one single agent responsible for a disease phenotype, there could be many bacteria work together to be responsible or aggravating or contributing to a human phenotype. And also it's a two-way street because some of the members may aggravate the disease, but others may actually protect us and benefit us. So in order to understand the causative role of gut microbiota in human disease and health, we need to start with the so-called microbiome-wide association studies. We need to have a platform where allow us to look at the variations of all the members of the gut microbiota and study the association with disease or health phenotypes. Try to identify all the members which are either positively or negatively associated with a particular disease. After we identify the potentially associated members, we should try to isolate them either into pure culture or into a consortium with defined membership. And then we should put these isolates in germ-free animals, being germ-free mice, germ-free piglets, or even germ-free non-human primates. But after you colonize the gut with defined members of human isolates, then you should try to apply the environmental condition which believed to trigger the particular disease and reproduce the disease phenotypes. If you can, and then you need to move to the molecular level, try to understand what kind of effect molecule produced by the colonizing gut bacteria interact with what kind of receptor from the host side, and trigger all kinds of signaling transduction pathway and change gene expression patterns, and eventually lead to the disease endpoints. You know, only after finish all these studies with such a high rigor, you may claim that you have identified members of the gut microbiota and understand their molecular effect, how to contribute to a disease or health phenotype. And then they can be used, both the bacteria and their effector molecules may be used as biomarkers as well as drug targets and for developing new therapy. So this is the thinking driving our own microbiome research. In order to start the microbiome-wide association study, and we use interventional longitudinal study design so that we change the microbiome by changing the diet. And then we see if there's any associated changes of host phenotypes. And then we collect the urine, blood, and fecal sample before, during, after intervention. And then using omics technology and multivariate statistics, try to profile all the variations in the three window and find the correlations among them and see if we can identify potentially important members of the gut microbiota and their effective molecule, and then move on to the mechanistic study. In order to do the microbiome research, we have several things in mind, and in all our microbiome studies. First, we understand that bacterial functions are strain specific. That means two strains in the same bacterial named species, they can share up to 30% of genomic difference. And this, we know that human and mice only share 10% genomic difference. So this means that under the same bacterial species, different strains can have a wide diversity in their genetics and of course, a wide diversity of their functions. In order to identify the truly important gut bacteria in disease or health, we need strive to go down to strain level. Otherwise, we may not be able to identify the key players. And we also need to keep ecology in our mind because the gut microbiota is a very complex microbial ecosystem. We know in macro ecosystems, members are not independent from each other. And members from very different taxonomic background can actually work together to form a functional group called guild. So members in the same guild, they work together, they thrive or decline together. But different guilds may compete or cooperate with each other. Then the question is, among the gut microbiota, who works with whom? And do they organize themselves also into such guilds? And another scientific ecological question is, in the macro ecosystems, not all the members are not equally important. And some members, for example, in the close forest, the tall tree species are the so-called foundation species, which structure and stabilize the whole ecosystem. If you lose the tall trees, you lose the whole system. And are there any members or any guilds of the gut microbiota which may also serve the role of the foundation species and to stabilize and structure the healthy gut microbiota? So these are the ecological questions we must answer when we do our microbiome study. And I would like to give you two examples in our own research focusing on obesity and type 2 diabetes. And we know metabolic diseases, obesity and type 2 diabetes are devastating epidemic throughout the world now. And there are many hypotheses about what drives such epidemic. So is it because we have taken too much of something bad, too much fat, or too much easy carbohydrates? Or is it because we have too little something good, and dietary fibers, for example? So our hypothesis is that it is probably driven by the lack of something essential, dietary fibers. And if you look at studies on fossilized human feces, coprolites of ancient human, you can realize that our ancestors probably had 200 to 400 grams of dietary fiber intake on a daily basis, while the average American may have only 15 grams. So over eons, our gut microbiota has been co-evolving with ourselves under a high dietary fiber context. And this changed only in recently, starting from 100 years ago in Western countries, and maybe 40, 50 years ago in Asian countries such as China. And interestingly, in traditional Chinese medicine, there is a tradition of using food as medicine. And if you look at those food, and many of them are rich in non-digestible polysaccharides, and you can also call them dietary fibers. And we collected a fecal sample from 314 young, healthy Chinese from 20 different locations throughout China, and 7 ethnic groups, half men, half women, half rural, half urban lifestyle. And after we analyze the microbiota, it's very interesting. There is an ethnic dependent segregation of the gut microbiota among the tested young, healthy adults. However, if you look at their gut microbiota, you can see that there are 9 genera short-chain fatty acid producing fiber utilizing gut bacteria, which make up the half of the total sequence. And these 9 genera shared by all the individuals, regardless of their ethnic background and the geographical location and the lifestyle and the gender. So this is the indication that healthy people's gut microbiota may be enriched by fiber utilizing short-chain fatty acid producing bacteria. And then this Lancet systematic review paper commissioned by WHO, they collected 135 million person years of data from 185 prospective studies, and also data from 58 clinical trials with 4,635 adult participants. So such a huge data set focused on the dietary fiber effect, quality of carbohydrates. And the bottom line is, regardless of the disease and disease types, there is a 15 to 30% decrease in all-cause and cardiovascular related mortality, and also decrease of incidence of coronary heart disease, stroke incidence, and mortality, type 2 diabetes, and colorectal cancer. So why high dietary fiber intake can have such a consistent and wide range of and benefits on a wide range of disease conditions? And we know that dietary fibers are non-digestible carbohydrates, but many of them are also fermentable. So that means they are complex carbohydrates. They escape the human digestion, but when they come to the gut, and they can become high quality energy source for many bacteria there. When bacteria ferment dietary fibers as energy source, they grow, but they also release a side product called short-chain fatty acids. And interestingly, hundreds of papers showed that short-chain fatty acids actually essential nutrients to human. And they have many functions, like provide energy to our colonocytes, mitigate inflammation, regulate satiety, but there's also an emerging pattern among studies of many chronic diseases showing that reduced abundance and diversity of short-chain fatty acid producing bacteria, and also lower production of short-chain fatty acids themselves seems to be associated with many diseases like type 2 diabetes and colon cancer. So that this also indicates that dietary fiber fermented by gut bacteria producing short-chain fatty acids may be an essential symbiotic pathway which for maintaining our health. And so based on all these understanding and the research over the years, we developed a dietary scheme where we included as much as dietary fibers as possible into the diet, and at the same time provide balanced macro and micronutrients to the host. So it's basically, it's a diet designed to feed the host, but also to provide energy to feed the gut microbiota. So we call it a feed my microbiome diet. And I was the first volunteer to try this diet, and I lost about 45 pounds over 1.5 years, and remained stable since then. It's been 10 years now. So it was reported as a feature news article in Science. If you're interested, you may read that article. And now I would like to share 2 particular studies and with you. One is high dietary fiber, excuse me, intervention of the gut microbiota among a genetic form of obesity called Prader-Willi syndrome. And children with this particular syndrome, has actually a genetic form of obesity. So they were born with very low muscle tone and even couldn't suck enough milk. Before weaning, they were malnourished. But after weaning, they quickly develop a form of hyperphagia, very severe. And they can never feel satisfied with any amount of food. And very quickly, they develop morbid obesity, like 5 years old, 50, 100 pounds. And this is a very difficult form of obesity to deal with. And however, in our hospitalized dietary intervention, targeting the gut microbiota with the high fiber diet. And we accidentally found out that about 30% of the morbidly obese children enrolled in the program turn out to be Prader-Willi syndrome patients. And also surprisingly, they showed a very good response to this microbiome targeted intervention. For example, this particular boy who was 140 kilograms at the initial, at the beginning, BMI over 60, and stayed in the program, hospitalized program for 285 days and reduced to 83.6 kilograms. And he continued the intervention at home. After 430 days, he was 73 kilograms. And by the way, all the participants in our programs had no extra exercise. And they only changed the diet. And so eventually we recruited two groups of morbidly obese children, one PWS and the other same morbidly obese, but with no genetic reason. And so again, 10% of initial body weight can be achieved in within 1 month in simple obesity children, and 20% weight loss can be achieved within 3 months for the PWS children. And they also showed improvement of lipid profile, glycemic response, and insulin sensitivity, almost all the parameters, and also showed improvement of the chronic inflammation. And so in order to show whether gut microbiota had any possible contribution to the improvement of the metabolic problem, we transplanted the microbiota from the same individual before and after the intervention. And interestingly, all the red colored data are from mice receiving the pre-intervention microbiota from humans, from the human donor, and all red colored data are from mice receiving microbiota from the same person, but after 3 months intervention. You can see that the pre-intervention microbiota induced first inflammation in liver and the gut, ileum and colon in the first 2 weeks. And after the inflammation was gone, they quickly started to excessively accumulate fat. They enlarge their adipocytes. And so basically the pre-intervention, but after intervention microbiota didn't have such effect. So the pre-intervention microbiota induced inflammation and fat accumulation, and post-intervention microbiota didn't. So this is an indication that at least the diet-induced changes of the gut microbiota contributed partially to the improvement of the metabolic problem. So in order to identify the key players which are responding to the dietary intervention and may play a role in the improvement of the metabolic health, we collected a fecal sample every month from all the individuals. And for each fecal sample, we sequenced 80 million reads in a high-seq Illumina high-seq platform, which equivalent to 8 to 10 gigabase pair of data. And so after the first round of assembly into contiguous, we recognized a little bit over 2 million non-redundant microbial genes from all of these samples. So this, from 109 samples, we generated 2 million genes. And each gene has an abundance in each sample from 0 to something. And so this is a huge data set with a lot of variables, but a very small number of samples. You need to reduce the dimensionality if you want to identify a pattern. Otherwise, it's almost impossible to analyze the data. But how can we reduce the dimensionality of such a huge data set? First, based on the principle of genetics, we know that genes which are from the same genome will behave exactly in the same way in the ecosystem. So based on this principle, we did a correlation among all the individual genes, and we found that more than there are 20,000 groups of genes. They behave exactly in the same way. And so we call them co-abundant gene group. Among such a co-abundant gene group, 376 containing more than 700 genes and many are typical bacterial genes. So these are potentially genomes of bacteria. And even though it's 376 genomes, they already amount to nearly 70% of all the sequence. So bacteria are the major members of the microbiome. And then we focus on the bacteria. But many of these bacterial genomes only show up in a few samples once. So we try to focus on 161 bacterial genomes, which show up in more than 20% of the samples. So they are the prevalent members of the gut bacteria. And so now we have 161 bacterial genomes, which makes up the 60% of the total gut microbiota. So they are the major members of the gut microbiome. And now we do the so-called co-abundant analysis among these genomes. And we know that the different bacteria may work with different bacteria to form a functional group called guild. So let's see who actually show up together with whom. So after doing this statistical analysis, we delineate the 161 genomes into 18 what we call genome interaction groups. And each can be considered also as a guild. So each colored group is potentially one guild. If two guilds connected with red line and they co-occur, so they increase or decrease together. But if they are connected with the blue line and they co-exclude from each other. And members in the same guild, they increase or decrease together. So we can add their abundance together, get the guild level abundance. So now we have abundance of 18 guilds. After we get 18 guild abundance, we try to correlate the guild abundance with many disease phenotypes. And interestingly, you start to see correlations. So 3 guilds show the negative correlation with many disease phenotypes, and 9 guilds show the positive correlation with many disease phenotypes, and the 6 guilds showed no correlation. So now we have mapped the major members of this particular, the gut microbiota of this particular tested human cohorts, and into 18 guilds. And we also have data to show potentially the 3 blue arrowed guilds. They are negatively correlated in their abundance with host phenotypes, so they are potentially beneficial. And the 9 red arrowed guilds, they are positively correlated with disease phenotypes, and they show they are potentially detrimental or pathogenic. And the other guilds, they showed no response, and they don't correlate with the disease, and they are potentially neutral. So in this way, we mapped the relationship among major members of the gut microbiota and also their relationship with the host. So this gave us a very firm background foundation to now develop a hypothesis-driven projects to understand why they are positively or negatively associated with the disease and what's the molecular mechanism. So this study is the first to show that bacteria in our gut may work as guild. But what kind of bacteria can make a guild? If you look at the members, the taxonomic background of members of each guild, you will surprise that members from different phyla can work together and in a guild. So this means that bacteria do not organize themselves in taxon, in genus, family, or or phylum, rather than to organize themselves into guilds. And also, even among the same species, you can see different strains can have different relationship with the disease. For example, we assembled 5 high-quality drafted genomes in this Eubacterium elegans, butyrate producing species, and we actually see 3 relationships with the dietary intervention. So positive responders, negative responders, and non-responders. So if you lump all the 5 strings together, you lose the actual picture of those members. So that's why taxon-based analysis. So analysis of your microbiome data based on taxonomy, based on genus, family, or phyla would not lead you to meaningful results. So please keep in mind that gut bacteria work as guilds rather than as taxa. And this is critical if you would like to discover truly important gut bacteria. Another research I would like to share with you is the high fiber dietary intervention among patients with type 2 diabetes. So basically, this is a randomized controlled study. We randomize type 2 diabetes patients into either UroCare or into the high fiber diet. But we control the calorie intake and macronutrient intake so that the only major difference in their diet between these two groups are the treatment group had nearly 40 grams of very diverse dietary fibers intake than the control group. And not surprisingly, both groups showed improvement. However, the high fiber group showed much more significant improvement in the primary outcome, HbA1c, and also in almost all the other secondary outcomes. And so we know that because of the randomized controlled design, we can only attribute the actual clinical benefit in the treatment group to the actual intake of dietary fibers. And dietary fibers are not digestible and cannot directly interact with human cells. So, but they can be served as energy source by some gut bacteria. So they have a potential to change the gut microbiota. And then we transplanted the microbiota from the same patient before and 3 months after the intervention to germ-free mice. Again, the before intervention gut microbiota from type 2 diabetes patients actually increased glycemic level. And but after intervention microbiota didn't. And so this is the indication, and also the microbiota from patients received the high fiber diet had much better glycemic control than the control group. So this is also another piece of causative evidence showing that the dietary fiber-induced changes of the gut microbiota may be mediating the benefits of the dietary fibers. And so then we collected a fecal sample each from every month from each individual. So now it's 170 fecal samples at 4 time points for all the individuals. And then we sequenced them and we assembled and identified a gene catalog of nearly 5 million genes. And we did the same thing. We did a co-abundant analysis among the genes, and we found 422 bacterial genomes. And they make up nearly 70% of the total sequence. So these are the major members. By using these 422 genomes to look at the structural variation, the global structural variation of gut microbiota during the responding to the dietary intervention, we can see that the two groups showed no difference in their gut microbiota structure at the baseline, but after 1 month, they both changed significantly from their own baseline, but also different from each other. However, after the first month, there's no further change. So this is the indication that this dietary intervention can indeed induce significant change of the gut microbiota, and it took about 1 month to finish that change. And after the 1 month, there's no further change. Because gut microbiota, because the dietary fibers, the way for dietary fibers to have any physiological impact on human body is by way of fermentation through gut bacteria and then produce short-chain fatty acids. So we profiled the changes of 6 short-chain fatty acids, and we found that 4 of them actually declined after the intervention. So that means they couldn't be responsible for the improved metabolic health. And then acetic acid showed no difference between the two groups, but maintained its level. So this, but butyric acid didn't increase in the control group, but significantly increased in the treatment group. So this is an indication that acetic acid may be mediating the clinical benefits shared between the two groups. However, butyric acid may be mediating the clinical actual clinical outcome only observed in the treatment group. And we know that acetic acid and butyric acid can bind to GPR43, GPCR43, and 4143 receptor in the L cell in our gut, stimulate GLP-1 production. And then GLP-1 can actually stimulate insulin production in the beta cells. And indeed, we see a significant increase of GLP-1 among the treatment group. And so this is an indication that the high intake of dietary fiber fermented into acetic, particularly butyric acid, may be the molecular pathway mediating glycemic control by increasing GLP-1 and then increasing insulin secretion. But if the story stops here, it's not surprising. And what is interesting to a microbiologist, to myself, is the reduced pH in the gut after the intervention. And we know that the pH is a physical, chemical, environmental factor which can affect the growth behavior of almost everything in the gut ecosystem. And so that we out of the 422 bacterial genomes, we assembled and identified 141 into high quality genome, and they also carry at least one short-chain fatty acid synthetic pathway. So they are genetically potentially short-chain fatty acid producers, 141, and they make up nearly 60% of the total sequence we generated. And among these 141 short-chain fatty acid producers, surprisingly, only 15 are positive responders to the high fiber intervention. So they become promoted by the dietary fiber. 79 remain non-changed, but 47 even declined, become lower than baseline. And so basically you have 15 on the left side, and they are promoted by the high fiber intervention, while 47 on the right side reduced significantly below baseline after patients take the high fiber diet. And so if you look at the bacteria on the left side, the 15 positive responders, they can produce, all of them can potentially produce acetic acid, 5 of them can produce butyric acid. While the members on the right side, which are reduced by the diet, you see many of them can produce endotoxin, which can induce inflammation. And some can produce endo. Some also can produce hydrogen sulfide. And these two compounds are known to be inhibitory to L cell for producing GLP-1. And interestingly, if we look at analyze the genetic capacity, genome of these positive responders and the negative responders, and we found that the positive responders, they have more genes for degrading plant polysaccharides. And while the negative responders, they had more genes in their genome to degrade animal carbohydrates, such as carbohydrates from mucin. So they have different preference to the energy source in their genetic capacity. And also the positive responders can produce more ATP and can generate more short-chain fatty acids, and they are tolerant in general to the low pH after they produce short-chain fatty acids and lower the pH of the gut. However, the negative responders, they actually are sensitive to low pH. So when that's how, after several weeks of competition, you eventually end up only a small number of positive responders, but the majority remain unchanged, but you also have quite a large number of the bacteria actually reduced. And fortunately, many of them are either toxin producers or pro-inflammatory potential pathogenic bacteria. So these are the list of these 15 strains. Some are very close to known members in our database. So they are well-known beneficial bacteria like Bifidobacterium lactobacillus and Faecalibacterium prosnitia and Eubacterium rectal. But others can only be identified to the genus level or order level or even other higher level. So that means they are novel bacteria. And we also showed that the total abundance and the diversity of these 15 bacteria. They are from 3 different phyla, but they work together as a guild. So we developed an active short-chain fatty acid producing index based on the total abundance and the diversity of this group of bacteria, and from each individual over different time points. And you can see that the higher this index a particular patient can achieve, the lower the HbA1c it eventually can reduce. And so in summary, surprisingly, a large amount of very diverse dietary fibers didn't increase the total diversity of the gut microbiota, but actually selectively promoted a small group of short-chain fatty acid producers and reduced the general diversity. And however, among the reduced diversity, you see pathogenic and detrimental bacteria. And so this is probably why, in addition to directly regulate the interact with host cells, there's also an ecological mechanism for this group of bacteria to benefit the host. And we start to isolate each individual strains from the 15 positive responders, like this one, so Bifidobacterium pseudocandidatum, showed the protective effect in high-fat diet-induced obese mice, and also in type 2 diabetes microbiota inoculated germ-free mice. And so this is another piece of evidence that this kind of bacteria, indeed, they are causatively contributing to improvement of the disease. So now our hypothesis is that this kind of positive responders can be considered as the foundation guild for a healthy gut microbiota or for a healthier gut ecosystem. And obviously, each individual has a different combination to different members to serve as the foundation guild. And some patients may have lost most, if not all, members of such a foundation guild. So you need to design a diet with different fibers which are fermentable by each person's foundation guild. And if the person is missing the foundation guild, you probably need to reseed from other healthy donors and then give the right nutrition to support its growth. And it also needs to grow to a certain level in order to change the whole gut ecosystem and exert health effect. So this probably is a new pathway for personalized nutrition. And so before we close, I would like to comment that short-chain fatty acids have been shown to boost antiviral immunity in mice. And also the ecological suppression of foundation guild through production of short-chain fatty acids to opportunistic pathogens such as endotoxin producing bacteria may also play a role in alleviating cytokine storm and sepsis. So those bacterial complications which happened very often in respiratory viral disease patients, for example, COVID-19. So there is a potential for this major symbiotic pathway to play a role not just in chronic diseases, but also in infectious diseases such as COVID-19. As a summary, I would like to comment that we need to have 4 perspectives in place in order to understand the role of gut microbiota in particularly interaction between diet, microbiome, and human health. First, always try to produce causality piece of data which can prove or demonstrate causality, even it's a small piece. And second, go down to a strain level or subspecies level in order to identify the true players. Third, understand that bacteria do not work in taxa, but work as guilds. So this is very, very important. Lastly, any analysis which can give you a good understanding and potentially give you high quality association between microbiome and human disease should be not dependent on the existing database. If you need the existing database to find the nearest neighbor to your sequence to be able to analyze your sequence, you will lose a large chunk of your data which has no near neighbor in a database, so that you throw them away and they don't participate into the downstream analysis. Obviously, this is a problem. So try not to use the database, start your microbiome data analysis. And you need to do non-targeted discovery analysis. Don't rely on any prior knowledge. Just look at the behavior of the sequence themselves, understand their ecological response and association with the disease. And then you may look at the database, and then you can tell which part of your sequence actually novel, which part of a sequence actually represents known bacteria. And that's how you should analyze your microbiome data. With that, I would like to acknowledge my students, my faculty members and collaborators, and also funding agency. And I'm happy to take questions.
Excellent. Thank you very much, Professor Zhao. Of course, we have a great number of questions. So just to start, one question is about dose. That is, what specific amounts and frequency of fiber intake might be needed to achieve the effects from a translation perspective that you have shown us? And does that need to be for a certain period of time as well?
Well, this is actually a fundamental question to understand how dietary fibers may change the microbiota, affect our health. And obviously, we still need a lot of work to be able to answer such questions. However, it looks like it takes about 1 month, over 28 days, for the foundation guild bacteria increase their abundance from this level to this level, and then they become stable. So this is the indication that it may need a few weeks for the new high fiber diet to stabilize a new structure of the microbiome. If you keep this new diet, you keep this new structure, and then the host start to recover from the previous disease. That takes time, and so this is. But then, what kind of composition would be needed for each person? You probably need to do this on an individual or personalized basis, because each person has different members, different kind of bacteria serve as the foundation guild, and different bacteria actually have different genes, carbohydrates, active enzyme genes, for them to be able to degrade and ferment different carbohydrates. So one dietary fiber, so the key is fermentable fiber. So we know some dietary fibers are non-fermentable, and so they just pass through the gut. They may be beneficial to the transit, but with very little other effects. So the most important fiber should be fermentable dietary fibers. And however, one particular fiber fermentable to my gut microbiota may not be fermentable to your gut microbiota, or has different efficiency for fermentation. So this is where we need to go down to personal level, or at least stratify them based on ethnicity group or geographical location, so that we can understand those details of your question.
Great, thank you. Here's a question that's specific to the type 2 diabetes study that you explained. Was that a crossover study, and how many participants were involved in each group?
Okay, so this is not a crossover study. This is a randomized 2-arm open label trial. And we have the two groups, we have about 53 participants in the first trial, in the mechanistic trial. But after we see the clinical benefits and the potential positive responders, which may mediate the dietary fiber effects, we organize another independent validation trial where more than 200 participants involved. And the same finding validated in that trial. So in this science paper, we published 2 trials, one small scale mechanistic trial and one larger scale validation trial.
Okay, great, thank you. Another question here is it known if acetate and butyrate have any negative effects on health?
And yes, yes or no. So basically, it's actually depending on the context. And for example, there's a research to show that acetic acid may be aggravating metabolic syndrome. However, the key piece of evidence is to infuse large amount of acetic acid into the stomach of the animal. We know that physiologically, acetic acid is not produced in the stomach. It's actually locally produced in the lower gut by bacterial fermentation. So if you infuse acetic acid in the upper gut, because acetic acid is so easily to be absorbable. So if you drink it, it's show up in your bloodstream almost immediately. Nothing can reach the lower part of the gut. So in addition to the direct beneficial effects through interaction with human cells, acetic acid and butyric acid also has a localized ecological effect if they are produced locally in the lower gut. And that's why, so if you infuse acetic acid through colon irrigation, you may see a very different picture. That is the protective and beneficial effects. So it all depends on, and also acetic acid can be produced from fermentation of carbohydrates, but also can be produced from fermentation of protein. However, when it's produced from fermentation of carbohydrates, and there's only short-chain fatty acid produced, sometimes also hydrogen, but there's nothing else. But if you ferment protein, in addition to production of acetic acid, you always produce some other toxic compounds like ammonia, amine, phenol, whatever. So because protein containing nitrogen and phosphate and sulfur. So you always have other detrimental compounds co-produced together with acetic acid. In that case, it's very difficult to tell them apart. Sometimes you see adverse effects of acetic acid. It's actually more probably coming from the other, detrimental compounds produced at the same time from fermentation of proteins.
Great. Okay. So this question is, you suggest that bacterial strain identification is key to understanding responses to diet, but we really only have probes for bacteria. Fungi, viruses, and protozoa might also contribute to metabolites that affect the human host. So wouldn't identification of functional genes provide a useful addition to inventorying the strains of bacteria? And this person's feeling is that both are critically important.
Yes, I agree. The microbiome is not just bacteria. You also have fungi, protozoa, and the virus, and of course. However, the most diverse, most abundant, and most metabolic active members are still bacteria. And I'm a bacteriologist, but it's my favorite study subject. But there's a reason that I focus only on bacteria. So one is the limitation of my own expertise. The other is it's actually the majority of the gut microbiome. And yes, if you do shotgun metagenomic sequencing from the 1 million or 5 million individual non-redundant genes, we know many of those genes are actually coming from non-bacterial members of the microbiome. But if we focus on the bacterial members of the microbiome, it's already 60–70% of all the sequence we generate. So that's why we just go ahead and analyze the bacterial part of the microbiome data. But this doesn't mean that bacteria are the only thing. Yes, we have approach, we have access to other part of the microbiome data that we can get access to, to do other analysis.
Great, thank you. I think we can take one more question, and just so others know, we will be sharing all the questions with Dr. Zhao afterwards. So the question is, and it's a little outside of what you spoke about, but is it possible to increase the positive guild population by using something like probiotics, so the positive guild could compete with the guilds that have a negative impact?
I would reframe the question into, can we use the members of the positive responders or the foundation guild as the next generation of probiotics? So if we know that probiotics is a very good concept, but the key is, how do you know the strain or the strains you use to produce your probiotics are ecologically competitive and also complementary in functions to the patient's gut microbiota? So basically, if the person has lost most or all the members of its original foundation guild, and then if you want to restore his or her gut ecology, you probably need to introduce similar members from other healthy donors, and then add the probiotics, and then give it the nutrition to support its growth. But I don't think you can just take a commercially available probiotics and give that to a patient without the scientific rigor to show that those probiotics, the strains used for producing those probiotics, actually can colonize the gut, complement the lost function. Unless you have the scientific data to show that, yes, it can. But there's a potential that the members of the foundation guild can be used as a new generation of probiotics.
Great, thank you, Dr. Zhao. That brings us to the top of the hour. So that concludes our webinar today. A reminder that the recording will be posted on our website, and we'll send everyone who registered a link to that. You can also find on our website information to register for the next webinar, and that is with Dr. Vanessa Ridora of Verily Life Sciences, and that's part of the Alphabet Group. So please join us again in a couple of weeks, and have a great day.